Now I'll be talking about how to read a clinical trial publication. Not how to read, but how I read, because I don't know what's the best way. This is the way that I do it. So this is how I read a clinical trial publication. And because I'm an oncologist and my field is oncology, I'll be citing examples from cancer drug trials, but I'm pretty sure that the principles will remain the same no matter which field of medicine you are in. And I'll be discussing a lot of concepts here. If you missed it, or if you want to read about those concepts in further detail, then I strongly encourage you to read this paper. It's open access. It will be available for free. And we have a beautiful diagram there that gives like a snapshot of the important things that you need to care for when you are reading a clinical trial. So I strongly encourage you to download this paper and read it. And you can also check out some of the talks that we have under the Queen's Global Oncology Program Masterclass, where we have several talks, but including this one that I gave on how to understand a phase 3 randomized clinical trial. So these will be complementary to what I'll be saying today. So, okay, how I read a clinical trial. So I get a phase 3 clinical trial report that I want to read. So what do? Even before I start reading the paper, I look at a couple of things that are of interest to me. The first is who funded the trial, who sponsored the trial. That is important to know because there is evidence to show that there will be lots of, you have to put your critical hat on, more so if it is an industry-funded trial, because there is data to show that there might be bias and spin in those reports. For example, this particular trial, this is from New England Journal of Medicine. Like all journals have their unique differences where they keep this information. But for example, New England Journal of Medicine has this at the end of the abstract about who funded it. So it was funded by this company, Roche Genentech. And in methods, they describe what the role of the trial sponsor or the funder was. So as you can see here, it says the trial sponsor collaborated with an academic steering committee, blah, 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 regarding trial design and data collection, analysis, and interpretation. And that is something that I don't feel very comfortable about. I have no problem collaborating with the industry for trial design, data collection, even analysis, but interpretation. I think the interpretation should not be done by the entity who has financial stake in the results. The interpretation should be left to the clinician investigators who are doing the trial. The interpretation should not be the role of the industry. But this is the reality. This is what is happening. I'm objecting to it, but this is what is happening. So you should at least be critical about it. You should watch out for this. Interpretation part, that I cannot be convinced at all that the sponsor or the funder should have any role at all in interpretation. That's like me holding, I don't know, a dance competition for kids and me being the judge and selecting my own daughter as the winner. And then the other thing that I look at is who wrote the paper, which is a pretty weird thing because you would assume that the authors wrote the paper. But in clinical trials, especially for clinical trials, that's not always the case. As you can see here, it says that all the drafts of the manuscript were prepared by the authors with editorial assistance from professional medical writers funded by the sponsor. Now, what is the level of that editorial assistance? That's the thing. In most of the cases, and I'm very saddened to tell you this, nowadays, it's not even this full paper. Okay, I'll understand somewhat. Okay, it's a full paper, probably. There are 25 authors, but none of them had the time to write the paper within the 5 years that they did the trial. Okay, but nowadays, even conference abstracts presenting at ESMO, it's like 300 words. Even those abstracts has acknowledgement for the professional medical writer. I'm like, if you do that for your thesis, you will be fired, right? Because that would be plagiarism. Or that would be ghostwriting. If you ask someone else to write a thesis, and that used to be the case. I used to see advertisements in Kathmandu in those electric poles saying, मात्र रु 10,000 मा मास्टर्सको thesis तयार, right? It's something like that. Like you pay certain amount of money and someone else will write it for you. And I am also interested in the affiliation of the authors, because as you can see here, some of the authors are direct employees of the industry. Again, that's not inherently bad because they contributed to the trial, but you should take all of this into account when you are interpreting the results of the trial. As you can see here, in the previous case, it said, in collaboration with an academic steering committee regarding trial design, data collection, analysis, and interpretation. Here it's blatantly the company was responsible for overseeing collection, analysis, and interpretation. This is even more suspicious in terms of how much you can trust especially the discussion part. And the manuscript was written with medical writing support, which was funded by the industry, with critical review and input from the authors. So clearly the authors did not write the first draft. They probably corrected a couple of grammars and spellings. This is a problem. This is a big problem. And this is one of the reasons why we are starting Common Sense Oncology, to highlight that we are starting to lose common sense in several aspects of oncology. So, okay, that's what I read even before I read the paper, because I want to know how much critically I should be reading the whole paper. Then the next question is, when do you read the abstract? So I read the abstract for two reasons. One is in order to gauge my own interest, whether I'm interested in the topic or not. So in order to know whether or not I have any interest in the topic, I casually read the abstract, and if it looks interesting, then I read the full paper. I don't make clinical or policy decisions based on reading abstract alone. That should not happen. You should not be making any important decisions based on abstract alone. Abstract is just to gauge your interest, whether or not it's worth downloading the full paper and reading the full paper. If it is something related to your clinical practice, then you should absolutely read the full paper. Don't make any clinical decisions based on the abstract alone. I also read abstract on the second occasion, that is, if once I have already read the full paper and I have already made an impression, but sometimes, I forget the data, and I have a patient in the clinic tomorrow or right now, and the patient is saying, okay, what are my chances of survival for 2 years if I take this drug? Now, I don't know exactly. It's 40%, 15%. I don't know exactly, but I have read the full paper and I know whether that drug is worth recommending or not. In that case, in order to quote those exact numbers, I quickly refer to the abstract and I can cite those numbers. So those are the two occasions in which I read the abstract. First, to decide whether or not to download the full paper. And second, once I have already read the paper, to just memorize a couple of data points. Okay, now let's go to the body of the paper. We have downloaded the full paper. You already know now what are the components of the paper and how they should be written. Now I'm talking more from the vantage point of a reader, not as an author, not as an editor or a peer reviewer, but as a reader. So the introduction talks about the rationale for the study. And because most of the papers that I read are from oncology, and I am already an oncologist and I am in the field, I usually know the rationale for the study in most cases. But yeah, introduction is very brief, and mostly it's about why this study needed to be done. And discussion is the interpretation of the study. And discussion is a part that it's okay even not to read. If you can critically read the data for yourself, you can decide how to interpret the data. But I'll tell you a couple of occasions in which I read the discussion very intently. So let's talk about the main crux of the paper, methods and results. So I'm highlighting only a number of points here because I don't have enough time to cover everything, and that's the reason why I recommended you a couple of resources at the beginning that you can refer to. So today I'll be discussing about these points. So first, let's talk about a phase of the trial. When you're reading the trial, you want to know what phase of the clinical trial it is. And there can be different phases: preclinical phase, phase 1, phase 2, phase 3, and then there can be phase 4, post-approval. So basically, phase 1 is a small trial done in limited number of patients to just check whether the drug is safe or not. If the drug is toxic, if it's not safe, then the drug's journey stops there. And if the drug is found to be safe enough, then it moves on to phase 2. There are always exceptions. I'm just giving you the classic story. In phase 2, we test the drug in a larger cohort of patients. And in phase 2, we are trying to answer the question, is the drug active? Is the drug, like in terms of oncology, we are asking, does the drug shrink the cancer? Is the drug even active? That's what we answer. And in phase 3, once we have decided in phase 2 that yes, the drug is active and it's shrinking the cancer, we want to know whether the drug is better than what we already have. If it's not better than what we already have, then there is no point in having it. So in phase 3, we do a randomized trial comparing this new drug with what we already have, called the standard of care. And if the drug is proven to be better, now we'll discuss later what that better means, but if the drug is proven to be better than what we already have, then it gets published in these big journals and gets approved by the regulatory agencies like the FDA, EMA, and so on, and gets reimbursed in countries with public health insurance system. But in many occasions, the drug will continue to be studied post-approval as well in a larger cohort of patients that are now eligible for the drug. And sometimes newer safety signals might also emerge. That post-approval studies are called phase 4. So to give you an example, this journey is quite tedious. It's not easy for a drug to reach from phase 1 to approval phase. Only 3.4% of cancer drugs that are tested in phase 1 ultimately get approved. And most of these drugs, they fail at phase 3 stage. So the probability of success at phase 3 is almost half, which makes sense because there is equipoise. And the cost of a cancer drug trial, it goes substantially higher as you move up from phase 1 to phase 2 to phase 3 because you are recruiting more and more patients. And therefore, a drug failing at phase 3 is a disaster, is a financial disaster. And therefore, you want to avoid that. And that's why I think it's even more important to be stringent at phase 1 and phase 2 so that we don't fail at phase 3. But the opposite is happening, unfortunately. So phase 1 trials, I have already told this, they ask the question, is the drug safe? But the problem is, most of the patients don't understand that. There have been several studies showing that patients who are getting enrolled in phase 1, they think they are going to benefit from the drug. When in fact, that's not even the clinical question there. The clinical question is just, is the drug safe? But most of the patients, they don't know that. 80% of the patients enrolled in early phase cancer trials were motivated by the potential of a clinical benefit, which is a big deficit on our part that we failed to educate the patients properly. Phase 2 trials, we're asking, does the drug work? Phase 3, yeah, we have already discussed this. So once we have decided that, okay, this is a phase 3 clinical trial, for example, then we need to look at what is the design of the trial. A trial can be a superiority trial, or a non-inferiority trial, or an equivalent trial. Most of the trials are superiority trials. So superiority trials are trying to see whether the new drug is better than what we already have. Non-inferiority trial is trying to see whether the new drug is not worse than what we already have. And equivalence trial is trying to see whether the new drug is within the margin of both worse and better, within an acceptable margin of what we already have. I'll not talk much about equivalence trial because they are rarely done. They are usually done in case of, for example, biosimilars. You want biosimilars to be equivalent to the original molecules. So, but in most of the cases, you don't see equivalence trials. So what are the superiority and non-inferiority trials? Fit for the newer drug. And if the hazard ratio goes over 1, that's worse outcomes with the newer drug. So in superiority trial, and that point is the midpoint of the hazard ratio, and the lines are 95% confidence intervals. So if the whole 95% confidence interval is less than 1, then that drug is superior to what you already have. Because even in worst case scenario, it is still beneficial than the standard of care. The second one, here you can see that there is this vertical line called margin. That is how you define the non-inferiority limit. That means, as I said, non-inferiority trials are testing whether the new drug is not worse than what we already have. So what does that mean? Not worse than. So that is what this margin is showing. And that is unfortunately consensus-based. People agree that, okay, if up to 20% more people die with this drug, then we will consider it non-inferior. We'll consider it acceptable. So that means that 95% confidence interval of the hazard ratio, if it crosses that 1.2, which is 20% increase in the risk of mortality, then it will be considered inferior. Otherwise, it will be considered non-inferior. So in the second row, you see that that whole line is within margin of 1.2. So we can consider that it is non-inferior. But it is also touching that line that passes through 1.0. So it is non-inferior. In the third case, it is again within the margin of 1.2, but the whole line does not touch 1.0 at all. So even in best case scenario, the newer intervention is worse than what we already have. So it is entirely non-inferior and inferior. And in the fourth case, where it has a huge range of confidence interval going from the benefit up until crossing the margin, it is inconclusive. You can't say anything about it. It could be better, it could be worse. And the fifth one, the last one, where it does not touch 1.0 at all, but also crosses that margin, it is completely inferior. So this is how we interpret from a non-inferior trial, whether the drug is non-inferior or inferior. So as you can see, sometimes even from non-inferior trial, you can conclude superiority if the whole margin lies below 1, as in the first row. So why do we even do these non-inferiority trials? I'm not talking about superiority because superiority is the most common type of trials. Your usual trials are superiority. Non-inferiority are exceptions. But why do we even do these trials? Why do we want to test whether the new drug is not worse than what we already have instead of testing whether it is better than what we already have? We do that on a couple of occasions. If the newer drug improves patients' quality of life, if it has lesser side effects, if it is less expensive, if it is less burdensome. For example, your standard of treatment for using, okay, let's take a real example. Let's say zoledronic acid. This is a drug that we use to prevent bone metastasis in patients with breast cancer. We used to give it every 3 weeks. And we wanted to test, is it too much? Can we reduce the frequency of the treatment? And then we tested whether giving it every 3 months instead of every 3 weeks has non-inferior outcomes. So it was a non-inferiority design trial because you would not expect that reducing the frequency would improve outcomes, right? At best, it will not worsen outcomes, but it will be less invasive for the patient, less burden for the patient, and less expensive for the patient. And that was a non-inferiority trial, which proved non-inferiority, and we changed our practice from using it every 3 weeks to using it every 3 months. But this thing is critical, the non-inferiority margin. How do we define how much worse is not worse? In the figure that I showed earlier, 1.2 hazard ratio, that is up to 20% increase in the risk of mortality, was considered non-inferior. But who decides that? Or is it actually acceptable to us? The patients might think that 20% increase in mortality is a huge risk. I don't want that. So defining that is critical. Because if that margin is too high, if you say the non-inferiority will define at 2.0, that means double the risk of dying, then everything will be non-inferior. It is very hard to make a drug that kills twice as many people. So, if it all depends on how you define not worse. So we did this study and published it in JAMA Open, in which we looked at the non-inferiority trials at that time that was published for cancer drugs. And we found that the range of non-inferiority definitions ranged up to the highest was 1.33. That means they were considering up to 33% increase in the risk of mortality as non-inferior. And as I said, in some cases, non-inferiority design we use to improve patient convenience, but we also found that in 40% of these RCTs, there was no reason to do a non-inferiority trial. There was no justification. The newer drug was not better in terms of quality of life, was not less invasive, was not less frequent, but still non-inferiority design was being used. But after we published this study, later on I came across an even more mind-blowing example of non-inferiority. So this is the trial. Can anyone guess what they defined as non-inferiority here? The limit of non-inferiority. What happened? Something happened, obviously. Okay, in the meantime, you can keep guessing what these boxes mean. I'll try to see if I can find this data online. But yeah, in this case, I'll come back to this example once I have it. Oh, no, okay, I could download it, no worries. So in this case, they defined non-inferiority at an upper limit of 1.8. 1.8. That means up to 80% increase in the risk of dying was considered non-inferior. And guess what? The trial was positive. They proved that pazopanib was non-inferior to doxorubicin. But what were the results? The hazard ratio was 1, and the 95% confidence interval went up to 1.53. But 1.53 is less than 1.8. So they considered that pazopanib was non-inferior. This is a really bad example. Like, it's a very good example, but this is a really bad trial in terms of defining non-inferiority. Okay, so after design, we should look at the eligibility criteria. What was the eligibility criteria of the trial? And nowadays, this is a big problem because there is a huge list of criteria for getting enrolled into a trial. Even reading all those criteria will take a long time. And so you can imagine that most of the patients, unfortunately, they don't qualify for clinical trials because they don't meet all of these criteria. This is just a random example. This is not anything particular. Most of the clinical trials are like this. But there will be very, very few patients who would hit all these restrictions and would be eligible for the trial, and that is a problem. There have been some efforts from organizations like ASCO to make clinical trials more inclusive and remove some of these restrictive criteria, but still, majority of the trials have all these restrictions. We did a study in which we looked at the phase 3 trials that actually led to drug approval. And even in those pivotal trials, there was less representation of female patients, less representation of black patients, less representation of very few patients with SBV and HCV infections, very few patients with brain metastasis. So they are being underrepresented. So these eligibility criteria are making sure that only the fittest of the fit patients are getting enrolled. And why does it matter? Like, why are we worried about it? Because first, that will mean that there is lack of generalizability of the results and confidence in the results for your patient population. Because the patient that you see in the clinic is not the patient who would have been eligible for the trial. So now you don't know whether the results of the trial will apply to your particular patient. And the other issue is the exaggeration of benefit. I'm giving you this example from an editorial that I wrote about hepatocellular cancer drugs. And in this table, you can see that the last 5 drugs are in second line of metastatic hepatocellular cancer. For second line metastatic hepatocellular cancer, the average survival would be less than 6 months. Even in first line, I think it's in the range of 6 to 8 months. But here you can see that even the control arm means no treatment. Even the patients who did not get any treatment are having average median survival of 7 to 8 months. So that means we are selecting the fittest of the fit patients, what Dr. Booth refers to as the Olympian patients, patients who would be fit enough to participate in Olympics despite having cancer. So there will be exaggeration of benefit. This benefit that we see in trials will shrink when you use these drugs in the real world. Okay, next, randomization and blinding. I think most of the, especially drug trials, they do a pretty good job about randomization. These are trials being done by these big pharmaceutical industries. And randomization is usually not an issue in modern trials, except in a few cases about unequal randomization. Instead of doing 1:1, sometimes they do 2:1, 3:1, and that would be a little too detailed to discuss here today. But randomization is usually taken care of. And blinding, I'll discuss about blinding later when I talk about placebo. Next, endpoint. So what's the primary endpoint of the trial? So the primary endpoint is what the trial is supposed to measure fundamentally and how the trial is powered and sample size is calculated. So it matters what the primary endpoint is. And usually for a patient with cancer, you would think that how long they live and how well they live are the two things that would matter to them. So in clinical terms, it would be referred to as overall survival, how long they live, and quality of life, how well they live. So that should be the clinical endpoints. These are the clinical endpoints, and they should be the primary endpoints. But there is something called surrogate endpoint. Surrogate means something instead of the real thing. So instead of the clinical endpoints, we are using something else that we believe will be a proxy for the clinical endpoint. And in oncology, we use response rate. That means what percentage of the patients have shrinkage of tumor. Progression-free survival. That means duration from treatment until the tumor grows beyond a limit. Time to tumor progression, disease-free survival. We have several surrogate endpoints in oncology. So why the problem nowadays in oncology is instead of using overall survival and quality of life as the primary endpoint, most of the trials are using these surrogate endpoints as the primary endpoint. And you might ask, like, why are we using that if they are not the clinical endpoints? The argument is using a surrogate endpoints will save time because you can measure tumor shrinkage earlier than waiting for the patient to die and see what the survival time is. And so it will allow earlier analysis, earlier publication, and earlier approval of the drug, which is true, but the difference is not as big as some people think it is. Because studies have shown that compared to using overall survival, if you use response rate, you save on average 19 months. And if you use progression-free survival, you save on average 11 months, which is important time saving, but it's not like 4 or 5 years that people think it is. But using surrogate would be fine if improving surrogate translated to improving survival and quality of life. But the problem is that's not the case. In most cases, the validation of the surrogate endpoints is pretty weak and pretty poor. That means improving PFS, progression-free survival, in most tumor types does not mean that the overall survival will improve. And that's where the problem lies. I'll give one example from breast cancer. So in breast cancer, these are the surrogate endpoints that are being used. But if you look at their correlation, pathological complete response does not correlate with OS. Response rate does not correlate. Progression-free survival, only mild correlation. Event-free survival, moderate correlation. Disease-free survival does not correlate, except in one certain subgroup. So in most cases, there is lack of good correlation between the surrogate endpoints and the real clinical endpoint. Okay, but survival is not the only clinical endpoint. There is quality of life as well. So does improving surrogate endpoint improve quality of life? Not necessarily. We studied that. You might think that delaying progression will help patients to have a better life, but we found that that was not necessarily the case because quality of life is affected not only by disease progression, but also by the side effects from the drug. And we found that the correlation was pretty weak between progression-free survival and quality of life. We published another paper last year looking into more details of this. And interestingly, what we found is if a drug improves overall survival, that drug at least does not worsen quality of life. At best, it improves. At worst, it maintains. But if a drug improves progression-free survival without improving overall survival, it could even worsen quality of life. Not just maintain, but it could even have detrimental effect on quality of life. And as I said, these are concepts that are relevant not only to oncology, but throughout medicine. In this study that we looked at COVID-19 clinical trials, we looked at the use of surrogate endpoints because for severe COVID-19, this is not just any COVID-19. For severe COVID-19, you would think that all-cause mortality and ICU stay would be the most relevant endpoints. But there were so many clinical trials measuring random things as the primary endpoint, like changes in oxygen saturation and long injury score, change in viral load, like all these random surrogate endpoints. Okay, after the endpoint, we should be looking at the sample size or the power of the trial, which brings me to this idea about statistical significance versus clinical meaningfulness. You all feel like what Archimedes felt when he discovered uh, and he started running around naked. Um, we feel that elation, that eureka moment, eureka, when we find this p-value of less than 0.05. You collected the data, you did everything, you analyzed, and finally when you hit that button and the p-value shows up as 0.03, you feel so happy. If it becomes 0.08, you're like, wow, can I maybe take 2 more samples and see if it becomes 0.049? So we all worship at the altar of p-value less than 0.05, which is unfortunate because I guess we did not talk about p-value at this workshop, but there is nothing magical about 0.05. And we should not lose common sense in interpreting this statistical significance. I'll give you one example from a breast cancer drug trial of neratinib plus minus capecitabine. This is the phase 3 NELA trial. In this trial, the drug improved median PFS by, is anyone good at maths? 5.6 versus 5.5. How much is that? 0.1 month. How many days is that? 3. It improved PFS by 3 days. What even is 3 days? If you forget that you have a CT scan appointment today and go next week, it will be 7 days. This drug improved PFS by 3 days and it got approved. Because look at the p-value. It's 0.0059. That's super impressive. But, okay, it is statistically significant, but is it clinically meaningful? Does it matter that our patients get a CT scan report today or after 3 days? It does not. So this is what we call statistically significant but clinically meaningless trials or clinically meaningless results. And why does this happen? The question is, why does just a difference of 0.1 month show up as such a significant p-value? That is what we call overpowering the trial. That means when you are planning the trial, you plan for a sample size to detect a minimum level of difference between the two arms. But if you include more patients than that, if you include huge number of patients, then any difference will turn up as significant. That's the reason, and that's what we call overpowering the trial, including more patients, huge number of patients to find anything as significant. Okay, let's talk about, and just to give you an example of how crooked the system is, this drug got approved. It did not improve overall survival. As you can see, overall survival did not improve at all. PFS improved by 0.1 month, but it still got approved. Next issue is the control arm, because this is a randomized trial. You have the drug arm and you have the control arm. So you have to think carefully about what the control arm treatment is. And the fundamental way of thinking about it is that control arm treatment the treatment that you would have used outside of the trial? If that patient was not a part of the trial, what treatment would you have used? And is that treatment included as the control arm? If not, then there is some issues with the control arm. So the control arm can be inappropriate if that is a treatment that is already proven inferior in previous trials. You already know that that treatment is worse. For example, the cemiplimab trial, they had a control arm of chemotherapy, when in fact, for this exact same population, pembrolizumab was already approved several years ago. And if patients did not have problem with access, they would have received pembrolizumab. But because they were a part of this trial, patients who got randomized to control arm, they got chemotherapy instead. And sometimes the trial does not allow an effective drug. And how do they do that? They say the control arm can be treatment of physician choice, whatever the physician wants to use. But there will be a but. And they say, but the choices should be among these 2 or 3 options. And the best option will not be included there. So you are forced to use an ineffective treatment. This is an example. In this, it says patients were randomly assigned to receive olaparib or treatment of physician choice. And in treatment of physician choice, they say you can use capecitabine, eribulin, or vinorelbine. But for these patients, we would have wanted to use carboplatin or cisplatin, but platinum is not in the option. So they don't directly say don't use platinum. They say you can use one of these three, and platinum is not there. And using a placebo instead of an active drug. So let's talk about placebo. Is using placebo inherently bad, especially in cancer? No. The answer is no. Methodologically, using placebo is the best scientific way to determine the efficacy of a drug, but it can be unethical if the standard of care is an active treatment. Sometimes it is even important to use placebo, and that is, I'll give you two examples of when you should be using placebo. Placebo is justified and in fact important if we are doing a trial of A plus B versus A. So A is the standard of care, B is a new drug, and you want to test A plus B giving together whether it's better than A alone. In that case, the trial should be A plus B versus A plus placebo. And the other is A versus no treatment, because the standard of care is no treatment. Like, let's say, active surveillance for low-risk prostate cancer. The standard of care is doing nothing. If the standard of care is doing nothing, then you want to test a new drug, then it has to be a trial of the new drug versus placebo. But this is unethical because this is a metastatic pancreatic cancer trial. And for metastatic pancreatic cancer patients, you don't put them on placebo. You put them on active chemotherapy. In this trial, they put patients on chemotherapy for 4 months, and then they just stopped it and gave half the patients this new drug and the other half no treatment, which is unacceptable. And even then, as you can see in this graph, PFS improved, OS did not improve. So even by giving control arm patients placebo, OS was not improved. And quality of life was reported as maintained, which means quality of life was also not improved. So this is the reality. Basically, this is a drug that does not improve how patient feels, quality of life did not improve, or how long patient lives, overall survival did not improve, but it will cost you more than $12,000 a month and has important side effects. Okay, the other issue is that of crossover and post-protocol therapy. What does that mean? That means, again, if you want to read about this in detail, I would encourage you to read this article. But what this means is the trial, like, let's say it's a trial of drug A versus drug B, and the patient will progress at some time from drug B. So once that control arm patients progress, are they allowed to receive the experimental agent? So in this example, osimertinib versus placebo. So once the patients in the placebo arm, once they progress, can they get osimertinib? That is the question. And this is becoming all the more problematic nowadays in adjuvant cancer trials. In this particular example, for instance, patients with this particular type of cancers, for them, osimertinib is the standard of care. They should be getting osimertinib when it is relapsed cancer. This is a trial of early stage cancer, before relapsing. And they are trying to see if you can give this drug before relapsing and it will improve outcomes. And it did, but the problem is the placebo arm patients, once they relapsed, they should be getting osimertinib, right? Because that's already the standard of care. But if you look at the supplementary table, only a few percentage of the patients, they got osimertinib when they relapsed. This is what we call crossover. The placebo arm patients should be crossovered to osimertinib, to the drug. Once they progress, they should be receiving the drug. But this lack of crossover will exaggerate overall survival benefit. Next, let's talk about censoring. What does censoring mean? So in a randomized trial, let's say you have a trial of drug A versus placebo or drug A versus drug B. There will be several patients in each cohort, in each arm. But sometimes these patients just drop out from the trial for different reasons. And if the proportion of patients that drop out from the trial is same in the drug arm and the control arm, then that is called uninformative censoring, because it's the equal proportion of patients who are being dropped out for random reasons. But if the dropout is more in one arm than the other, then you will have to think why this is happening, and it is called differential or informative censoring. So what that fundamentally means is, let's say it's a trial of drug A versus drug B, or drug A versus standard of care. And if the drug A is toxic, for example, has several side effects, then the patients might be dropping out from the trial due to those side effects. Then essentially what we would be doing is, when we are comparing the patients in the trial, if we forget these patients who are being dropped out, then we are comparing only the fit and healthy patients who are still staying in that drug arm with all patients in the control arm. So that would be a biased interpretation. Again, if you want to read in detail about this, I encourage you to read this second paper that I have put here, which is based on the interpretation of the first paper over there. So you should be reading both these papers. And essentially what we show in this article is, these are the Kaplan-Meier graphs. And over here you can see in the top row, without adjustment. One is for DFS, the other is for overall survival. But what you can see is, if you don't adjust for that censoring, you will see a biased result. You see an impressive hazard ratio, you see a highly significant result. But if you control for that, if you assume that patients who are being differentially dropped out are being dropped out because they have progressed, then that benefit disappears. It narrows. So the benefit does not look as impressive. So this is a little tricky concept to understand, especially if you are not even in the field of oncology, but this is something that happens in cardiovascular trials as well, the issue of informative censoring. And yeah, this is, I would say for you, it's not must know, but it is good to know, fancy to know concept. But one thing that is absolutely important for you to know is intention to treat versus post-protocol analysis. What does that mean? Let's imagine drug X versus best supportive care, a randomized trial of adjuvant lung cancer. Let's say we'll randomize patients after surgery, and we'll randomize them to receive 1 year of drug X versus best supportive care after they have completed their chemo. Let's say we enroll 2,000 patients. We randomize them 1:1. So 1,000 patients in the control arm, 1,000 patients in this new fancy drug arm. Of the 1,000 patients in the control arm, 610 died. Try to remember these numbers. 610 died. And now there are 1,000 patients in the drug arm. Of those 1,000 patients in the drug arm, only 300 could complete the whole course of treatment, of which 15 died. Of those 300 patients who completed the total treatment, 15 died. About the remaining 700 that did not complete treatment, let's say 595 died. So what does per protocol analysis mean? Per protocol means you will analyze only those patients who received per protocol, the exact whole treatment that you wanted to give them. Per protocol, what the protocol says. So in this case, control is, of course, 1,000 patients, 610 died, so 61% died. But if you look at the drug arm, only 300 patients completed the protocol treatment. So you are taking only those 300 patients, and only 15 of them died. So it's like only 5% died. The drug seems to be magic. We don't have any drug like this in oncology. 61% of control arm patients died. With the drug, only 5% died. Wow, blockbuster. But this is wrong analysis. You cannot do per protocol analysis for this reason. It will give you very flawed result. There is something called as treated analysis, which is even worse. If there is an analysis that is even worse than per protocol, it's called as treated. As treated means you will analyze based on what actually happened. So in this case, because those 700 patients did not complete the treatment, you will put them into the control arm. And so the control arm became from 1,000, it became 1,700. So you included those 595 patients from those 700 who died into the control arm death as well. So you came up with 71% death rate with the control arm. And with the drug arm, it's still 5%, 15 of 300 died. So now your drug looks even more impressive. If you can complete 1 year of your drug, only 5% will die. Otherwise, 71% will die. But what is the correct analysis? It's called the intention to treat analysis. It does not matter what happened. What matters is what you intended to do when the trial is started what was supposed to be. So in intention to treat analysis, you include the whole 1,000 patients that were randomized to the active arm. And if you look here, of those 1,000 patients, 15 plus 595, 610 died with the drug as well. So in fact, it's exactly the same percentage of the patients who died in either arm. 61% died in either arm. So in fact, this drug is as good as placebo. It does nothing. But if you looked at per protocol and as treated analysis, this drug looks like a magic bullet. So you should always do intention to treat analysis. There are some exceptions. Let's not go into exceptions today. But by definition, by default, ITT analysis is the analysis to do. Because what you want to know is the effect of your decision. You can't make decisions retrospectively. So you are making decision today. Should I treat this person with this drug? Should I not treat this person with this drug? Should I take path A? Should I take path B? After you go to path A, if a tiger eats you, that does not matter. It all should be accounted for within the intention to treat analysis. So by definition, always do ITT. And again, in fact, one of my favorite persons on Twitter, he does not tweet lately a lot, and I miss his tweets. He's very funny. You can follow him. He has several tutorials, and he's very funny, as you can see. He says intention to tweet. And so he will give you a lot of Twitter-based education. Okay. Yeah, I think I'm running out of time, so let's not talk into detail of this. But this is where you see how many patients are dropping out and what's happening. The first figure 1 of every clinical trial. In that flow diagram, you will see how many patients were randomized and how many patients were randomized to each arm and how many patients started dropping out from that for different reasons. A couple of words about subgroup analysis. This is a classic example that I want to cite because this trial had changed treatment guidelines, but this was based on subgroup analysis. So classically, we are taught that for non-small cell lung cancer, if it is non-squamous histology, you have to use pemetrexed. If it is squamous, use gemcitabine. That's a very strong recommendation from most of the guidelines. But it was based on this particular trial, and these were the results. So the left side, it favors CP, which is cisplatin plus pemetrexed. Right side, CG is cisplatin plus gemcitabine. But if you look at all the subgroups that they have done, almost everything is touching the middle line. That means there is no big difference, except for these couple of lines that are barely touching it. And they are highlighting this. Non-squamous is adenocarcinoma. Non-squamous, pemetrexed seems to be better than gemcitabine, p-value of 0.03. Squamous, gemcitabine seems to be better, p-value of 0.05. So this changed the guidelines. And I was very disappointed to see this because, first of all, in this forest plot, the way you check whether there is important differences between subgroups exist or not is actually you draw a vertical line through all patients, the first one. All patients, the middle point, you draw a vertical line, this red line that I have drawn here, and you see whether all these horizontal lines touch this or not. If they touch, there is not much subgroup differences there. And the other one is, over here, there are 19 subgroups. You can't just keep testing in different subgroups. You have to control for that, and you have to pre-specify them. If you do it post hoc, that does not have any weight. You have to pre-specify what subgroups you are going to see. And this paper, it's not the fault of authorship, but the paper clearly mentions that the p-values were not adjusted for multiple comparisons. They say that they did not adjust it, but the guidelines changed. If they had adjusted for this, if they had done a correction for this multiple comparisons, only a p-value of less than 0.0026 would have been considered significant. But they did not adjust for it, p-value of 0.03, 0.05. These changed the guidelines. And yeah, after knowing this, I had written this paper calling it the billion-dollar subgroup analysis because the sales of pemetrexed based on that subgroup analysis started to rise. Yeah, let's talk about toxicity description. This is the last point. So whenever you are reading the conclusions or the abstracts of a clinical trial, you will come across this type of language called manageable and mostly reversible safety profile. there will be something like, some trials will say acceptable toxicity. Some will say the treatment was feasible. Someone says favorable toxicity profile, well tolerated. But these are just subjective terms used to downplay the harms. You should look at the numbers yourself. You should not just trust this language. I'm citing this example because, this is so interesting. It says mostly reversible. What do you understand by reversible? It means you can reverse the side effects, right? Manageable, you can manage the side effects. But if you look at the exact data, fatal adverse event was 5 versus 0. That means 5 people actually died from side effects. If someone died, how can you reverse that? By definition, it's not reversible. Yeah, and if it was manageable, you could not, they would not have died. Just a couple of examples to make you aware of these critical appraisal scales. This trial conclusion says regorafenib should be considered a treatment option. So you would think that this is a really good treatment. If you look at the results, PFS was 3.6 versus 1.7 months. 2 months improvement in PFS. Okay, 2 months improvement in PFS. But if you look at the OS, it was 11.1 with the drug and 13.4 with placebo. So patients with the drug had shorter overall survival than patients with placebo. And if you look at toxicities, okay, let's not go into this. I don't have much time. Okay, let's talk about discussion. So when do I read discussion? I read them sometimes to discover this type of biases in, because I do this type of research. So it interests me to know how much of bias is there. So I read that, and sometimes I read discussion to answer some methodological questions. Sometimes I'm interested, why did they do this? Why did not they do this? And usually in discussion, they usually have answers to those questions because the peer reviewers will also probably have asked those questions. And usually discussion is the place, if you have a question, oh, why did they not do this? Or why did they use this treatment instead? They will usually have answers in the discussions. And most important is the limitations. They will usually have a limitation section where they describe the limitations of the trial. And a final word about guidelines and editorials, because once the trial is published, it goes into guidelines and there will be editorials written about it. You should not take them at face value. These all slides will be available on the website, so you can read these papers in detail later. But the gist here is there will be lots of conflicts of interest in the guideline, and there will be lots of over-interpretation. So don't trust guidelines, and neither should you trust editorials, because people who are getting money from the same company are writing the editorial about the same drug. And so they will have a biased interpretation. So don't trust editorials, don't trust guidelines, because now you know how to read a clinical trial by yourself. So trust your own self. Again, these are other multiple examples of how experts are always wrong. And sometimes they say that the onus is to prove that the drug is not helpful. It's the other way around. Don't let them convince you otherwise. The onus is to prove that the drug helps more than it harms. It's not the other way around. It's just like this case, right? The guy says, I ordered a lamp from you, but I never got it. And that person says, can you show me an evidence? And he just shows an empty hand. Like, what can there be an evidence of not getting something? That brings me to common sense oncology. I described several examples using oncology trials, several problems, and therefore we wanted to improve this. And with the leadership of Dr. Booth here, we have started this movement called Common Sense Oncology. I encourage you to follow our Twitter handle, as well as go into our website if you are interested in oncology, to see how you can participate and what is happening in Common Sense Oncology movement. Thank you very much.